My name is Coral, and today we're going to be talking about point mutations. Mutation is the result of a substitution of one base for another. And there are two types of point mutations. Transitions happen when a purine is substituted for another purine, or a pyrimidine is substituted for another pyrimidine. Transversions occur when a purine becomes a pyrimidine, or vice versa. Point mutations can have one of the four following effects: missense, nonsense, silent, or read-through. Let's dive a little bit deeper to understand what each of these effects are. First, let's start with our original DNA sequence. We can group these into codons and use our genetic code table to find the corresponding amino acids. Now let's say we have a transition happen in alanine. Now we have an A replacing the G, and this changes the corresponding amino acid in the sequence. Instead of alanine, we have threonine. This is an example of missense. The point mutation causes a change in one amino acid, but doesn't affect the rest of the sequence. Let's go back to our original sequence. What if this time we had a transversion? In this case, we have an A replacing a C. This results in a stop codon being formed instead of cysteine. This means we truncated our sequence too early. When this happens, it is known as nonsense. This is because a codon that codes for an amino acid is changed into a stop codon. Again, let's return to our original sequence and see what happens when we have a transition happen somewhere else in the sequence. Now, our codon would have been AAT instead of AAC, but lucky for us, these two codons code for the same exact amino acid, so no real change occurred. This is what is known as a silent error. The mutation causes a change that codes for the same amino acid. We have one more type of error to discuss, so let's see what happens when we have another transversion occur. Here, we have a TGT instead of a TGA. Instead of having a stop codon, we now have another cysteine. But wait, it doesn't stop there. Without the stop codon, the sequence will continue, tacking on proline and glycine onto this protein. When this happens, it is known as an error called read-through. This means the stop codon was mutated into another amino acid that results in a longer protein. Well, there you have it. Those are the 4 errors on point mutations that you need to know. Tune in next time to learn some more genetics. Thanks.